Welcome to the O-Levels Hub. It's O-Levels Biology 5090 lesson, Chapter 5, Enzymes. 5.1 Enzyme Action. 5.2 Effects of Temperature and pH on the enzyme activity. Definition. Enzymes are proteins that function as biological catalysts and are involved in all metabolic reactions. What are catalysts? A catalyst is a substance that increases the rate of the chemical reaction and is not changed by the reaction. Substrate. A substrate refers to the specific molecule or compound upon which an enzyme acts. The substrate binds to an enzyme's active site, forming an enzyme-substrate complex. Once bound, the enzyme catalyzes a chemical reaction that converts the substrate into one or more products. Characteristics of enzymes. They have high catalytic power and can quickly catalyze a chemical reaction with a tiny quantity. They accelerate reactions while remaining unaltered during the process, means they are unchanged at the end of the process. Temperature, pH, and inhibitors can all have an impact on enzyme performance and function. Enzymes are often quite specialized, catalyzing only one type of substrate. Lock and key hypothesis. According to the hypothesis, like a lock can be opened by its key only. A substance possessing specific composition only can combine with the specific active site found in the specific enzyme surface. The molecules on which the activities of the enzymes take place are called substrates. The substrates possessing accurate geometric configuration can fit within the enzyme's active site. The enzyme's active site is highly specific to the surface of the substrate. The substrate and the enzyme fit like a lock and key. Hypothesis. Providing a lock and key enzyme action model. This is the image from which you can get a better concept of lock and key hypothesis, in which the lock acts as an enzyme and the substrate acts like a key. And when the substrate fit in the active site, they are only specific substrate that fit the specific enzyme's active site. Effect of temperature and pH on the enzyme action. Enzymes are proteins and have a specific shape. Very important. Held in place by bonds. This is extremely important around the active site area, as a specific shape is what ensures the substrate will fit into the active site and enable this reaction to proceed. Enzymes work fastest at their optimum temperature. In the human body, the optimum temperature is 37 degrees Celsius. Heating to high temperatures beyond the optimum will break the bonds that hold the enzyme together and it will lose its shapes. It is known as denaturation. Substrates cannot fit into denatured enzymes as the shape of their active site has been lost. Denaturation is irreversible. Once enzymes are denatured, they cannot regain their proper shape and activity will stop. For example, this is an enzyme and this is the active site of the enzyme. Amino acid chain making up the enzyme means this is the chain, means proteins. Forces between amino acids in the chain hold the enzyme together and create its unique shape. If we increase the temperature, see its active site has changed and the enzymes are denatured. Now the substrate no longer fits in it, so the reaction will not happen. Increasing the temperature from 0 to the optimum increases the activity of the enzymes, as the more energy the molecules have, the faster they move and the number of collisions with the substrate molecules increases, leading to the faster rate of reaction. This means that low temperatures do not denature enzymes, they just make them work more slowly. Effect of temperature on enzyme function. This is the graph which is very important and this often comes in the exam. At low temperatures, fewer successful collisions, so enzymes plus substrates have less kinetic energy. So low activity and substance, less substrate turned into products. See, these are low. And if we increase the temperature, more successful collisions will happen as the kinetic energy is more in the molecules, so they will collide with each other, and enzymes plus substrates have more kinetic energy, so more substrate turned into product. And then it is the optimum temperature, means the temperature at which the enzymes work its best. At temperature above optimum, more collisions, but active site has been deformed. Collisions will increase, but the active site has been denatured or deformed, so enzymes will no longer act as it was doing at its optimum temperature. Effect of pH on enzyme action. The optimum pH for most of enzymes is 7, but some that are produced in acidic conditions such as the stomach have a lower optimum pH of pH 2, and some that are produced in alkaline conditions such as duodenum have a higher optimum pH 8 or 9. If the pH is too high or too low, the bonds that hold the amino acid chain together to make up the protein can be destroyed. This will change the shape of their active site, so the substrate can no longer fit into it, reducing the rate of activity. Moving too far away from the optimum pH will cause the enzyme to denature and activity will stop. This is the active site and the complementary shapes. Substrate, if we increase the pH far above the optimum, the shape of the active site will be changed. So the substrate will no longer fit into it and it will not catalyze the reaction. The graph of the pH means at the extreme pH furthest from the optimum, enzymes are completely denatured, means no activity over here. And it's the optimum pH, means 8 is the optimum pH of this enzyme. Remember, this depends on where the enzyme works in the body. Not all the enzymes have 8. Some may have 2, some may have 5, some may have more than 8. Moving away from the optimum pH results in gradual loss of the activity as active site has become distorted or deformed.